So today we are going to discuss about mechanism of hormone action. That how hormones used to act on their target cell. When we discuss about the definition of hormone, what are hormones? These are chemical messengers. They are intracellular and they are produced in what amount? Trace amount. So the concept would be that hormones has been divided into two categories. There is one category which is lipid soluble and there is other category which is lipid insoluble, or let's call that water soluble. Which hormones are lipid soluble? All the steroid hormones. If I ask you to give me some example of steroid hormones, all the hormones of adrenal cortex, they are steroid. For example, aldosterone. Second example is cortisol and sex corticoid. Moreover, the hormones secreted by glands, testes and ovaries, testosterone, estrogen, progesterone, these all are steroid. And steroid hormone, as we know that steroid hormones are derived from cholesterol and they are lipid soluble, so they can easily pass through plasma membrane. Apart from steroid hormone, there is one more category of hormone which are lipid soluble, and that category is iodothyronine. And which are iodothyronines? T3 and T4. And what about the third hormone of thyroid gland? And what is the composition? TCT is a protein hormone, right? Water soluble, they are not lipid soluble, they are not allowed to pass through the lipid membrane. And all the hormones, either they are amino acid derivative or they are any example of amino acid derivatives? Any example? Melatonin is a derivative of? Melatonin is a derivative of serotonin. Which amino acid? Tryptophan. Answer is tryptophan. And derivative of tyrosine is? Adrenaline, noradrenaline and dopamine. All the polypeptide and proteins, you can take the example of insulin, glucagon, और अभी ये बताई थी PTH, TCT, moreover जो भी हार्मोन्स हमने पढ़े थे डाइजेस्टिव ट्रैक्ट में, फॉर एग्जांपल सिक्रेटिन, गैस्ट्रीन, एंट्रोगैस्ट्रोन, एंट्रोकाइनिन, CCK-PZ, all the hormones are protein in nature. So basically, by structure, hormone could be lipid soluble or lipid insoluble. Let's say this is cell here, and the cell is covered by a living membrane. What we call that? Plasma membrane. And what plasma membrane is made up of? Phospholipid. Which is the most abundant phospholipid there? Lecithin. Lecithin is the most abundant phospholipid. If I take a section of plasma membrane, you will find out that there is some part of plasma membrane which is water soluble, water loving, or there are certain part which are water hydrophobic or water hating. लिपिड बाइलेयर होता है, लिपिड सिंगल लेयर होता है, बाइलेयर. Similarly, hydrophobic end and hydrophilic end. This is how plasma membrane is. If we go in detail, these parts are fatty acid, and fatty acids are attached with glycerol, and glycerol is attached with phosphate, and phosphate is having choline. So this here is choline. Choline is attached with phosphate. Phosphate is attached with glycerol, and eventually there would be fatty acid. So these fatty acid, they provide what nature? Hydrophobic, and the rest part is hydrophilic. So if I am taking a plasma membrane here, there can be two conditions. Either the molecule are lipid soluble, so they are allowed to go inside the cell, or the lipid molecule or the molecule is lipid insoluble, they are not allowed to go inside the cell. So eventually a hormone, as you are telling me, it's a chemical messenger, or it need to control or it need to influence the metabolic and physiological action of the cell. The metabolic and physiological nature of the cell can be influenced or can be changed by two ways. Either hormone is allowed to go inside the cell or hormone is not allowed to go inside the cell. This is something like, that's a school, and there are two types of student. One, the student which is having admission in school, paying all the fees, so allowed to go inside the cell, and second student which is not having admission in the fees. So first what would happen? This student need to meet gatekeeper, then need to meet receptionist, then other principles, and then eventually go to the class. So those which are not allowed to go inside the cell, they would be acting by that mechanism. Alright, so first we will be learning the mechanism of water-soluble hormone, that how water-soluble hormone act. And there are certain receptors which are present inside the cell. What would be the name of receptor? Intracellular receptor. And there are certain receptors which are present outside the cell. They are extracellular receptors. So these hormone, the water-soluble hormone, they are not allowed to enter inside the cell, and they act on extracellular receptor. Who will tell me the definition of receptors? Transducer proteins. That's great. And they act on intracellular receptors. And most commonly, see, these receptors could be in cytoplasm, they could be on the nuclear membrane. So most commonly, these receptors are present within the nucleus. 90% of time they are present within the nucleus. And what is contained inside the nucleus? Yeah, nucleus is like a tijori, and all the genetic material is contained inside the nucleus. So they would be influencing the expression of gene, and they would be controlling the regulation of physiological and metabolic activities. We will be learning that. So have we done about the basic concept? So basically hormones are of two types, and mechanism of action would be of two types. Either they are going inside the cell or they are not going inside the cell. So let us start about the mechanism of hormone action, or mechanism of those hormones which are going inside the cell. Okay? So let us first discuss the mechanism of action of those hormones which are not lipid soluble. First we will be learning a basic concept, that how hormone act. You know something called a secondary messenger. So the basic concept would be that this is a plasma membrane here, and within that plasma membrane, okay, when you study the structure of plasma membrane, it consists of phospholipid and certain proteins, and what we call that intrinsic and extrinsic protein. So there are certain integrated proteins which are present within that plasma membrane, and proteins can be enzymes, say yes or no? Yes. Right. So there are certain enzymes which are present here, and other molecules which we will be learning, and these enzymes for the time being they are not active, these are inactive enzymes. And there is a protein which we call G protein. Ever heard about G protein? Right, G protein is a transducer protein, or transducin we call that, and we call that that's a transmembrane protein. So what is the basic concept about G protein? G protein is having how many subunits, how many domains are there? 3 domains. And what are the name of those domains? Alpha, beta and gamma. So alpha, beta and gamma are 3 domains of G protein, and when they are not regulating any action, what is attached to them? GDP, guanosine diphosphate. So when these proteins are not performing any action, they are not activating or inactivating any enzyme. They are not, these are signaling protein as well. So when they are not under signal, they are inactive and they are attached to GDP. And with this G protein, there is a transducer protein, another transducer protein which is receptor attached to it. For example, in this membrane, there is a G protein here, alpha, beta and gamma. And what would be attached? GDP. It's inactive right now. And to this, there is a protein which is like this, and this protein is extracellular receptor. So that's your extracellular receptor. That's your G protein, alpha, beta, gamma unit, and these are some inactive enzymes. So what happens whenever a molecule which is not allowed to go inside the cell, it binds here, it acts as primary messenger, and certain conformational change occur. You know, there is something which we call phosphorylation. What is your concept of phosphorylation? Addition of phosphate. So when this receptor is activated by this hormone, this protein undergo phosphorylation, and when it undergo phosphorylation, GDP would be converted into GTP. And when this protein is charged now, addition of phosphate make the protein charged. After charging, there is a breakdown, and alpha along with all the GTP goes away, and beta and gamma remains there. And this alpha along with all the GTP activates other enzyme. And in the cytoplasm of cell, there is ATP which is present there, there is GTP which is present there, and other molecule. The activated enzyme transform or convert this ATP into cyclic AMP. So cyclic AMP is a secondary messenger. GTP into cyclic GMP. Other molecule like inositol triphosphate, DG, calcium, they all act as secondary messenger. So the basic concept is a hormone come and bind, and binding of this primary messenger result in conformation of certain change in the protein part of membrane, and certain enzymes are activated. This enzyme result in the generation of secondary messenger, and these secondary messenger initiate enzyme cascade, initiate the activation of certain enzyme. When one enzyme is activated and it is going to activate the other enzyme, we call it enzyme cascade. For example, we all are standing in a line and I am not able to communicate with the person who is the first. So what I am doing, I am tapping the shoulder of next one, then next one would be tapping the shoulder of next one, and so on. So a cascade has been initiated. Or if anyone is missing, the cascade will not be initiated. Like in glycolysis, what you have studied, that one enzyme, 10 reaction 10 step reaction is there, and when one enzyme metabolize a substrate, then a product is formed which act as a substrate for the next enzyme. You got my point? So similar thing would happen here, that these secondary messenger initiate the activation of a cascade, and we will be learning example of one enzyme, how example of one hormone, that how this cascade is activated. तो आपसे क्या पूछा जाएगा, आपसे एक बहुत कॉमन सा सवाल आएगा कि इनमें से कौन से सेकेंडरी मैसेंजर हैं. तो ये 5 सेकेंडरी मैसेंजर आप बहुत कॉमनली याद रखेंगे. ठीक है, प्राइमरी मैसेंजर कौन होता है, सेकेंडरी मैसेंजर कौन होता है, ये याद रखेंगे, और सेकेंडरी मैसेंजर का जनरेशन कैसे होता है वो याद रखेंगे जो हम पढ़ने वाले हैं. डन अप टू हियर? एनी क्वेश्चन और डाउट? नो क्वेश्चंस? ऑलराइट. Okay, when it come to hormone, can you tell me the one of the most commonest hormone which act on our cell and initiate the response? Let's say that hormone is involved in the sympathetic responses. What would be your answer? Epinephrine or second adrenaline. So let us learn how adrenaline work. So to understand this, I need to make a plasma membrane here, skeleton of a plasma membrane, and just ignore it that I am making it very rough. And I will be making certain molecules here. Okay, what is the effect of adrenaline? If I am having a rush of adrenaline right now, what will be the effect on my glucose, blood glucose? Sympathetic nervous system initiate rise in blood glucose or decrease in blood glucose? There would be rise in blood glucose, na? So other cell are able to use glucose. I need glucose right now or not? If I am in sympathetic action, I need glucose actually, right? So how adrenaline molecule initiate the rise of blood glucose? और जब rise of blood glucose होगा तो कैसे होगा? क्या मैं फटाफट खाना शुरू कर दूंगा? ये जो stored glycogen है वो ब्रेक डाउन होकर आएगा. तो ग्लाइकोजन को कैसे ब्रेक डाउन करता है? ग्लूकोज में. नाउ डोंट टेल मी कि सर ये तो काम हमने पढ़ा है ग्लूकागोन का. ठीक है, एडेनोसिन एक्ट ऑन देयर, हाउ इट इनिशिएट द रिस्पॉन्स वो पढ़ लेते हैं. ओके, यू टेल मी व्हाट इज दिस हियर? This is an external receptor, right? And to this external receptor there is a protein, transmembrane protein. The name is G protein, and right now it is inactive. That's GTP or GDP? GDP. GDP is also associated with some economic condition of our country. हाँ सर, ये सुना है, बहुत बहुत नीचे है आजकल है. Okay. Now there is a hormone here. And this these are couple of hormone. There are so many hormones which are associated here. Right now I am very focused on this man, and he is very unhappy. Why is he unhappy? Because inactive right now. And what's his name? Adenylate cyclase. It's inactive adenylate cyclase. There would be guanylate cyclase as well, जो अलग न्यूट्रॉन्स अलग हार्मोन में रिस्पॉन्स करेगा. So a molecule of adrenaline comes here, and this is primary messenger or secondary messenger? Primary messenger. और जैसे रिसेप्टर पे बाइंड करेगा क्या होगा? ऑटो फॉस्फोराइलेशन, और there would be addition of a phosphate to here. So this GDP would be transforming into what? GTP. And when this G protein is charged, there is a cleavage here. And this beta and gamma remain membrane bound, and alpha along with all the GTP, it runs away and it goes and activate this enzyme. And when adenylated cyclase is active, what it do? The primary function of this enzyme, that it transform molecule of ATP into cyclic AMP. So by the activation of this enzyme, what has been generated here? Secondary messenger. Then what would activate it now? There would be activation of enzyme cascade. This cyclic AMP activates a kinase enzyme. You know what are kinase enzyme? Yes, shabash. Any enzyme which is involved in the transfer of phosphate or addition of phosphate, we call that kinase enzyme. There is a protein kinase, and protein kinase are simply abbreviated as protein kinase A, B, C, D and so on. So there is a protein kinase A. Remember, for adrenaline is a A. Oh, easy way to remember, for cyclic AMP it is A. So protein first cyclic AMP activate प्रोटीन काइनेज ए. इसके साथ साथ हम एम्प्लीफिकेशन भी ऐड करेंगे यहाँ पे कि कैसे एम्प्लीफिकेशन होता है, कैसे एक हार्मोन का मॉलिक्यूल मिलियंस ऑफ सबस्ट्रेट मॉलिक्यूल बना सकता है. तो जब साइक्लिक एमपी बना तो उसने क्या किया, प्रोटीन काइनेज को एक्टिवेट करना शुरू किया. जब प्रोटीन काइनेज है वो एक्टिव होने के बाद दूसरे इनएक्टिव एंजाइम को एक्टिव करेगा. देयर इज ए इनएक्टिव एंजाइम, द नेम इज फॉस्फोराइलेज. सो इनएक्टिव फॉस्फोराइलेज इज एक्टिवेटेड. और जब फॉस्फोराइलेज एक्टिव हो जाता है, फॉस्फोराइलेज ये एक्टिव करता है दूसरे एंजाइम को, उसका नाम होता है, दिस इज आल्सो इनएक्टिव, ग्लाइकोजन फॉस्फोराइलेज. और जो ग्लाइकोजन फॉस्फोराइलेज है जब वो एक्टिव हो जाएगा, नाउ द रियल स्टोरी वुड स्टार्ट. फॉस्फोराइलेज it would be acting on glycogen. और ग्लाइकोजन फॉस्फोराइलेज जब ग्लाइकोजन पे एक्ट करता है तो बनता है ग्लूकोज 6 फॉस्फेट. और जब ग्लूकोज 6 फॉस्फेट से फॉस्फेट रिमूव हो जाएगा तो बन जाएगा ग्लूकोज. और ये ग्लूकोज कहाँ जाएगा, सेल से बाहर. और सेल से बाहर क्या है यहाँ पे, ब्लड वेसल. तो जब एडेनिन मॉलिक्यूल गया सेल के अंदर, उसने पहले जाके क्या किया, जी प्रोटीन एक्टिवेशन किया फॉस्फोराइलेशन से. जी प्रोटीन का जो एक यूनिट है वो जाके किसको एक्टिवेट करता है, इनएक्टिव एंजाइम को. इट कुड बी एनीथिंग, इन अवर केस इट इज एडेनाइलेट साइक्लेज. एडेनाइलेट साइक्लेज एक्ट करता है एटीपी पर, वो एटीपी को कन्वर्ट करता है साइक्लिक एमपी में. साइक्लिक एमपी एक्टिवेट करता है प्रोटीन काइनेज को, जो फॉस्फोराइलेज ग्लाइकोजन फॉस्फोराइलेज एक्ट करेगा ग्लाइकोजन पर, ग्लूकोज 6 फॉस्फेट बनेगा एंड इवेंचुअली बाय लूजिंग 1 फॉस्फेट इट वुड बिकम ग्लूकोज एंड लेवल ऑफ ग्लूकोज इज इंक्रीज इन ब्लड. सो ये जो यहाँ से यहाँ तक काम है, इसको क्या बोलेंगे हम, व्हाट इज दिस कॉल्ड एज एंजाइम कैस्केड. एंड व्हाट इज दिस कॉल्ड एज सेकेंडरी मैसेंजर. एंड व्हाट इज दिस कॉल्ड एज प्राइमरी मैसेंजर. ये तो एक सिंपल सा एक्शन थ्योरी पढ़ लिया. अब पढ़ते हैं एम्प्लीफिकेशन. व्हाट इज एम्प्लीफिकेशन. एम्प्लीफिकेशन को अगर आपको डिफाइन करना पड़े आप कैसे कहेंगे कि सर अगर इफेक्ट कुछ गुना बढ़ जाए तो हम उसे कहेंगे एम्प्लीफिकेशन. एनी आइडिया ग्लूकोज के कितने मॉलिक्यूल रिलीज होंगे ब्लड में बाय द 1 मॉलिक्यूल ऑफ एड्रेनालिन. एक होना चाहिए. अब कितने होते हैं वो पढ़ते हैं. तो हम यहाँ ना एम्प्लीफिकेशन लिखना शुरू कर देते हैं. कितना मॉलिक्यूल आया मेरे पास हार्मोन का, कितना मॉलिक्यूल आया हार्मोन का, एक. और एक मॉलिक्यूल एक रिसेप्टर पे बाइंड करेगा. एंड दीज रिसेप्टर्स आर वेरी 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 स्पेसिफिक. इनको मैं एड्रेनर्जिक रिसेप्टर कह दूंगा. नाउ एक जीटीपी एक्टिवेट करता है 10 एडेनाइलेट साइक्लेज को, तो इफेक्ट कितना गुना हो गया. और एक एडेनाइलेट साइक्लेज एक्टिवेट करता है 10 एटीपी को, तो कितना हो गया 10⁻². एक साइक्लिक एएमपी एक्टिवेट करता है एक प्रोटीन काइनेज को, तो कितना हो गया अभी 10⁻². और एक प्रोटीन काइनेज एक्टिवेट करता है 100 फॉस्फोराइलेज को, तो कितना हुआ 10⁻⁴. और एक फॉस्फोराइलेज एक्टिवेट करता है 100 ग्लाइकोजन फॉस्फोराइलेज को, कितना हो गया अब 10⁻⁶. और एक ग्लाइकोजन फॉस्फोराइलेज एक्ट करता है 100 ग्लाइकोजन मॉलिक्यूल पर, तो कितना हो गया 10⁻⁸. तो कितने ग्लूकोज के मॉलिक्यूल निकलेंगे? मिलियंस ऑफ ग्लूकोज मॉलिक्यूल वुड बी। ठीक है, तो जो हम पढ़ते थे एम्प्लीफिकेशन ऑफ सिग्नल, इसको बोलते हैं एम्प्लीफिकेशन ऑफ सिग्नल, जब एक मॉलिक्यूल एक्टिवेट होकर मल्टीपल मॉलिक्यूल को एक्टिवेट करे। तो क्विक रिवीजन करें एम्प्लीफिकेशन का, कितने प्राइमरी मैसेंजर? 1। और कितनी जीटीपी एक्टिवेट होगी? कितनी जीटीपी एक्टिवेट होगी? 10 और 1? हाउ मेनी? 1 जीटीपी, 1 जी प्रोटीन वुड बी एक्टिवेटेड ना। और अल्फा अलोंग विद ऑल द जीटीपी, ये कितने एंजाइम को एक्टिवेट करेगा? 10 को। सो 1 जी प्रोटीन वुड बी एक्टिवेटेड एंड एम्प्लीफिकेशन वुड बी 10 टाइम। और ये कितने एटीपी को कन्वर्ट करता है? 10 को। तो 10 कितना करेंगे? 100 को। तो कितना होगा? 10 टू द पावर 2। अब ये वो फेज आता है जब 1:1 का रेशियो चलता है कि एक साइक्लिक एएमपी एक ही काइनेज के लिए बना है। तो 100 साइक्लिक एएमपी 100 काइनेज, इससे रिमेन 10 टू द पावर 2। फिर एक काइनेज 100 फॉस्फोराइलेज, एक फॉस्फोराइलेज 100 ग्लाइकोजन फॉस्फोराइलेज, और एक ग्लाइकोजन फॉस्फोराइलेज 100 ग्लाइकोजन मॉलिक्यूल, and glycogen molecule then make the equal number of glucose 6-phosphate and glucose. So amplification is up to 10 to the power 8. It occurs in a 1 or 2 minutes. It does not take very long time. So you do not need much molecule of adrenaline. You just need few molecules of adrenaline in order to get the effect. On the target cell, it should not be like adrenaline. Achha, hormones are used in the action or they remain unused? They are used in the action and eventually they are degraded by किडनीज एंड लीवर, राइट। तो ये एक एग्जांपल हो गया। अच्छा, यहाँ पे एक और चीज़ ऐड कर देते हैं, हम पढ़ के आते हैं एंटागोनिस्ट का इफेक्ट। यू नो व्हाट इज द मीनिंग ऑफ बीइंग एंटागोनिस्ट? अपोजिट। ग्रेट, यू आर सो स्मार्ट। इफ दिस हार्मोन इज एसिटाइलकोलिन हियर, then the effect on cell would be exactly opposite. Yes or no? Acetylcholine would also undergo autophosphorylation, but this time which enzyme would be activated? Guanylate cyclase. This time this said boy, it would be activated guanylate cyclase. And what would be the target substrate? ATP or GTP? GTP. And GTP would be converted into cyclic GMP. Now the secondary messenger is cyclic GMP. So if cyclic AMP is produced, a type of effect is produced, and cyclic GMP is produced, other type of effect would be produced. All the enzymes would be inactivated. So how hormones act antagonist? They act antagonist by producing different type of secondary messenger or different type of response enzyme cascade inside the cell. It makes sense? We are very clear about it. Second, we will be learning the example of secretin. The next we will be learning. अभी हमने कितने secondary messenger cover कर लिए? 2, cyclic AMP and cyclic GMP. अभी बचे हमारे पास IP3, कैल्शियम एंड डीजी। एक एग्जांपल में वो भी पढ़ लेते हैं, और फिर इंसुलिन कैसे काम करता है वो पढ़ के देन सेकंड ग्रुप ऑफ हार्मोन को डिस्कस करेंगे। डन अप टू हियर? एनी क्वेश्चन और डाउट? यस और नो? ठीक है। सेकंड, लेट अस डिस्कस दैट हाउ अदर सेकेंडरी मैसेंजर्स आर जनरेटेड। एंड नेक्स्ट हार्मोन विच आई एम गोइंग टू पिक इज दैट हार्मोन विच वाज फर्स्ट डिस्कवर्ड. You know who is the guy who discovered the first hormone? Any idea? Name of those? Yes, yes. Say it loudly. Starling and Baylis, right? And which hormone they discover? Secretin. Okay, secretin is protein and estradiol. Protein. And what is the role of secretin, by the way? From which part of body secretin is secreted? Duodenum. Okay. And what is the role?
बाइकार्बोनेट ऐड करके देता है।
कहाँ पे? बाइल जूस में।
और उसको स्टमक,
मुझे लगता है बाइल जूस कम फ्रॉम लीवर, पैनक्रियाटिक जूस कम फ्रॉम पैनक्रिया। तो वेयर इज द साइट ऑफ एक्शन ऑफ सिक्रेटिन? कहाँ से ऐड करता है? कहाँ से लेके आता है बाइकार्बोनेट? फ्रॉम द ब्लड। तो फ्रॉम द ब्लड इट ऐड to the pancreatic juice, and it also act on the stomach. And what it do? All right. So again, this is plasma membrane, and these are the receptors for secretin, and these associated protein, again alpha, beta, and gamma, and right now inactive GDP. This time the molecule which we are going to learn, there is another inactive molecule और इसका नाम होता है PIP2. PIP2 is phosphatidyl inositol 4,5 biphosphate. So there is a molecule which is phosphatidyl inositol 4,5 biphosphate, PIP2, right? Now a molecule of secretin or digestive hormone, it come and bind here, that's primary messenger, and this molecule undergo autophosphorylation. The GDP would be converted into GTP, and then there would be cleavage here, and alpha along with all the money, all the GTP, where it goes and activate it. And when this molecule is activated, it cleaves into two components. There is one component which is formed by its cleavage, it is IP3. IP3 is inositol triphosphate. And the second component which remains membrane bound is DG, and DG is diacyl glycerol, right? So by action of alpha along with the GTP, PIP2 is cleaved into two secondary messengers, inositol triphosphate and diacyl glycerol. Major secondary messenger जो concerned होगा वो होगा DG. So what is the role of IP3? IP3 is present in cytoplasm and it is freely water soluble. So this IP3 moves from here and goes to a cell organelle, and that cell organelle is the storehouse of calcium. Can you predict the name of this cell organelle? Endoplasmic reticulum. Which part? Terminal cisternae. And there is a channel here, and this channel is having receptor for IP3. So right now calcium channels are blocked, and calcium are willing to go outside, but they are not allowed to go outside. So this IP3 comes and bind here, and after binding of IP3, what happens? Calcium come in the cytoplasm. So how many secondary messengers are in the scene now? There are three secondary messengers which are in the story now. First is IP3, second is diacyl glycerol, and third is calcium. This calcium along with this DG, see DG activity, diacyl glycerol activity is dependent on calcium. There are certain protein kinase, we call it protein kinase C. Protein kinase C are inactive, and they can be only activated when they are catalyzed by DG in presence of calcium. You know, enzymes need certain cofactor for the activity, and cofactor could be metal ion. So this diacyl glycerol need metal ion to activate protein kinase C, and after activation of protein kinase C, the cascade would initiate, and there would be other enzymes which has been activated. For example, pyruvate dehydrogenase is also activated. I am not going in detail here, and you don't need to remember that. And there would be physiological effect. And what would be the physiological effect? क्या बता रही थी आप? Bicarbonate would be added from the blood to pancreatic juice, and it would be inhibitory effect on the stomach as well. So we have discussed the action. After the effect, what would happen? Reverse reaction would start. This calcium would come back here. There are certain calcium holding protein. You must be knowing the name of few. Calciquistrin. You know calciquistrin. And the calcium is there, IP is released. And IP goes back to DG. Hey DG, where you are? I was missing you so much. Let's again get. There is a patch up, right? And when IP3 is combining with DG, what would form? PIP2. And when PIP2 is formed, what would be detached? GTP, and it has used its phosphate, and alpha is having GTP now, and alpha would be going back to beta and gamma, and now they would be making a inactive molecule, and they are waiting again for another molecule, right? So this is how other secondary messengers are generated. There is one more hormone which is very common. You know, the most potent anabolic molecule of my body is anabolic. अच्छा ana भी बोली है. insulin is the most potent anabolic molecule. She is very right. And how insulin works? Insulin is a protein. How many amino acids are there? 51 in active form, right? And how many chains?
2.
1 and 2, right? That's A and B chain to be exact. That's A and B chain. Alpha and beta chains are present in hemoglobin to be exact. Remember that? Alpha 2 and beta 2. The insulin is having, A chain is having 21 amino acid and B chain is having 30 amino acid. Anyways, cell like liver cell and adipocyte, they are having receptor for insulin. And when we talk about insulin receptor itself is a protein domain. And the protein domain is something like this. These receptors are paired and they are having two parts, alpha unit and beta unit. There are two subunits of insulin receptor. One subunit is alpha unit and second subunit is beta unit. And this beta unit is an enzyme. And name of this enzyme is tyrosine kinase. You got my point? So when insulin molecule come and bind here, what happens? There is autophosphorylation of tyrosine kinase. It's like they have been charged. And once they are charged, they activate the other insulin receptor in the cell. There is a complete reception system which we call insulin reception system, IRS. And insulin reception system is activated, then the cascade would start. We don't need to go in detail, just remember that the insulin receptor is having how many subunits? Alpha 2 and beta 2. So don't get confused that insulin chains are A and B, and alpha 2 beta 2 are the receptors, and these are two receptors. One receptor have one alpha and one beta, and each beta chain is an enzyme. Which enzyme? Tyrosine kinase. And by the binding of insulin, they have been autophosphorylated. So you have discussed two different conditions of diabetes mellitus. The first condition is type 1, when beta islet of Langerhans are destroyed and they are not making enough insulin. So we call that insulin dependent diabetes mellitus. The second, and that generally occur, the symptoms generally occur before the age of 20 years. And diabetes mellitus type 2, which is insulin independent diabetes mellitus. In that condition, what happens? Sufficient of insulin is produced, but there is a conformational change in this receptor. And insulin is binding and there is no autophosphorylation and the reception system is not activating. So if you are having type 1 diabetes, you might be having some insulin injections because you are not having insulin. In type 2 diabetes, you are making insulin, but cell cannot utilize insulin. And you need certain other molecule which can enhance the formation of this receptor and so on. We are not going in detail there. So that is the example of those hormones which act by extracellular reception. ठीक है. बेसिकली अगर नीट लेवल की बात करें, आपसे ना एक कैटेगरी पूछी जाएगी कि कौन सा हार्मोन सेकेंडरी मैसेज जनरेट करेगा, कौन सा नहीं करेगा, तुम आराम से आंसर कर दोगे. अगर उसने ये भी पूछ लिया कि इस हार्मोन से कौन सा सेकेंडरी मैसेंजर जनरेट होता है, तो आप वो भी आंसर कर देंगे. एड्रेनलिन एंड एसिटाइलकोलिन के क्लियर है, साइक्लिक एएमपी एंड साइक्लिक जीएमपी एंड सिक्रेटिन, और जितने भी डाइजेस्टिव एंजाइम्स हैं वो किस मैकेनिज्म से काम करते हैं? डीजी, IP3 एंड कैल्शियम. ठीक है बेटा. सी वी हैव लर्न वन मोर रोल ऑफ कैल्शियम. Calcium is so important to us. Most abundant mineral of our body. Answer is calcium. ठीक है. Now, second category of hormone which are freely allowed to go inside the cell. How they work? बस वो पार्ट पढ़ लेते हैं. ठीक है. फिर I will be telling you about antagonist and synergistic effect. ठीक है. So on the other hand, there are certain hormones which are freely allowed to enter inside the cell, and they act on intracellular receptors which are mostly nucleocytoplasmic. Intracellular receptors are mostly nucleocytoplasmic. Wow. How much percent are nuclear? 90% are nuclear. ठीक है. So they would be acting on nuclear receptor and they would be initiating the physiological response. When you read your text book, ये वाला diagram है text book में. ठीक है. So this is the membrane here. Let's say this is the ovarian membrane. And there is a hormone coming here. Which hormone is mentioned there? Estrogen. See, spelling same होता है, either O without O. ठीक है. Estrogen goes inside the cell very freely, and the receptor could be here, and that is a nuclear membrane, or the receptor could be here, and this is what the genetic material is. You have something called transcription. What is transcription? Making a copy of RNA from DNA. A transcript which has been encoded in form of nucleotide, and we call that messenger RNA. And these hormones, they act as a key, and this key, when bind to the receptor, this key can open transcription or this key can block transcription. So we used to remember that these act as transcription factor, and when this DNA is transcribed after the binding of hormone, DNA is transcribed, and what would be formed? RNA. And when this RNA is translated, what would be formed? Protein. So by this same thing, this thing is known as central dogma or gene expression. So which hormone control gene expression? Which hormone control gene expression? Answer would be steroid hormone or iodothyronine, those which act on intercellular receptor. So they undergo inside the cell, act on special nuclear receptors, work as transcription activator, transcription factor, and can induce a gene expression. And after the formation of that protein, what would be there? There would be, let's say, a physiological effect or physiological response would be there. एक वर्ड होता है कलेक्टिव और क्यूमुलेटिव. क्यूमुलेटिव का मतलब ये हुआ हार्मोन आया, हार्मोन ने आगे बाइंड किया रिसेप्टर पे, रिसेप्टर ने इनिशिएट किया ट्रांसक्रिप्शन, ट्रांसक्रिप्शन के बाद ट्रांसलेशन हुआ, प्रोटीन बनी. उस प्रोटीन के बाद क्या आया? फिजियोलॉजिकल इफेक्ट. तो क्यूमुलेटिव बायोकेमिकल इफेक्ट रिजल्ट इन फिजियोलॉजिकल रेस्पॉन्स. क्यूमुलेटिव और कलेक्टिव biochemical effect result in physiological response. तो अगर दो तरह के सवाल पूछे जाते हैं, पहला क्वेश्चन, ये कौन से हार्मोन जीन एक्सप्रेशन को कंट्रोल करते हैं? योर आंसर वुड बी स्टेरॉइड एंड आइडोथायरोनिन. और कौन से क्यूमुलेटिव और कलेक्टिव बायोकेमिकल इफेक्ट से फिजियोलॉजिकल रेस्पॉन्स आता है? आंसर शुड बी दोज विच आर गोइंग इनसाइड द सेल. तो कौन कौन, अच्छा, इफ दिस इज FSH इज मेड अप ऑफ व्हाट? यू नो, ओवियन मेम्ब्रेन हैव रिसेप्टर्स फॉर, इट्स अ ग्लाइकोप्रोटीन. तो FSH होगा तो FSH वुड बी बाइंडिंग हियर एंड सेकेंडरी मैसेंजर वुड बी जनरेटेड एंड कैस्केड वुड बी देयर. इफ देयर इज एस्ट्रोजन, देयर इज प्रोजेस्टेरोन, इट इज टेस्टोस्टेरोन और अदर्स हार्मोन, इट कुड बी अदर सेल, cortisol could be there or aldosterone could be there. We have discussed about aldosterone in renal physiology. So what I have told you, aldosterone act on which cell? DCT and collecting duct. और DCT पे याद है कुछ सोडियम पोटेशियम टू क्लोराइड पंप बताया था, और एल्डोस्टेरोन के आने से कैसे उसकी परमिलिटी बढ़ जाती है. ठीक है, दैट इज अबाउट योर इफेक्ट ऑफ हार्मोन और मैकेनिज्म ऑफ एक्शन ऑफ दोज हार्मोन विच एक्ट बाय विच मैकेनिज्म? इंट्रासेलुलर रिसेप्शन. अब हार्मोन ना दो तरह के हो सकते हैं बच्चे. हार्मोन हो सकते हैं आपके एंटागोनिस्ट और हार्मोन हो सकते हैं आपके साइनेजेस्टिक. साइनेजेस्टिक इफेक्ट का मतलब क्या होता है? सेम टाइप. कुछ हार्मोन बताओ जो साइनेजेस्टिक इफेक्ट करते हैं. ईस्ट्रोजन, प्रोजेस्टेरोन, प्रोलैक्टिन एंड ऑक्सीटोसिन. इनका क्या साइनेजेस्टिक इफेक्ट होता है? ये दोनों चाहिए होते हैं डेवलपमेंट ऑफ मेमोरी ग्लैंड. ड्यूरिंग द प्रेगनेंसी व्हेन देयर इज डेवलपमेंट ऑफ मेमोरी ग्लैंड, which hormones are responsible? Estrogen and progesterone. Then prolactin is milk producing hormone और oxytocin is milk ejecting. इस पूरे प्रोसेस को बोलते हैं लैक्टेशन. तो लैक्टेशन में मेमोरी ग्लैंड के एल्वोलाई भी डेवलप होने चाहिए जो ईस्ट्रोजन प्रोजेस्टेरोन के इन्फ्लुएंस में डेवलप होंगे. प्रोडक्शन ऑफ मिल्क भी होना चाहिए जो प्रोलैक्टिन करेगा, ऑक्सीटोसिन. तो ये यहाँ पे कैसे काम करते हैं? सिनर्जिस्टिक. किसमें? लैक्टेशन में. आगे चल के आप पढ़ोगे प्रोलैक्टिन हैव नो गुड रिलेशन विद एस्ट्रोजन एंड प्रोजेस्टेरोन. तो इफ वी आर हैविंग हाई लेवल ऑफ प्रोलैक्टिन, इवेंचुअली एस्ट्रोजन एंड प्रोजेस्टेरोन लेवल वुड बी गोइंग लो. तो जब तक आपका एस्ट्रोजन प्रोजेस्टेरोन डेवलपमेंट करे तब तक प्रोलैक्टिन कम रहेगा. जब प्रोलैक्टिन डोमिनेंट हो जाएगा तो एस्ट्रोजन प्रोजेस्टेरोन इफेक्ट ऑन मेमोरी ग्लैंड कम हो जाता है. ठीक है, बाकी जगह काम करता रहेगा. दैट इज अबाउट द mechanism of hormone action. Any question or doubt about this topic? Are we 100% clear? Fine. Lecture is completed. Thank you.